light does half of the job, and then the other half is done by food.
Right.
So you also pointed out another thing. You brought up the example of stress hormone cortisol. Cortisol is regulated by circadian clock, but at the same time, we know it's a stress hormone. So if you have stress in the middle of the night when cortisol is not supposed to be high, it will not just wait for the morning time. Your cortisol will go up, and we know that. So similarly, the master clock sends the signal that when it should be day, when we should be eating, and when we should be fasting. And accordingly, the liver clock, the gut clock, all these clocks, they time themselves. And when I say time themselves, what happens is, just imagine there is you are kind of driving in a downtown area with a lot of stoplights and green lights, right? And if you imagine if there was no light anywhere, then there will be a lot of accidents, or there will be a lot of traffic congestion. So having the lights done on and off at the right time help the traffic move. Similarly, in our liver, if you imagine we are eating a lot of different type of food that has to be metabolized, that has to be broken down, sorted out, then a lot of things that we don't need for our body, for example, the artificial sweetener, the coloring agent, the flavoring agent, all of them go to another conveyor belt. They get excreted out, and the protein gets broken down, and they build up other part of the cell. So a lot of things are going on at that time. So it's almost like you're bringing in food, dividing them into different parts, and moving through this conveyor belt or traffic. So not everything will happen at once. So there is time for protein to break down, there is time for glucose to be made, there is time for nucleotide to be made, there is time for bile acids to be made, different hormones to be made. So the clocks actually help time different things. And when clocks break down, then what happens is you can imagine either a traffic jam or a big pileup. So the metabolism is not efficient anymore. There is a lot of byproducts just lying around, and that stresses the cell, and then we get to the disease. So my point is, although we have clocks, these clocks also respond to when food is coming in. At the same time, when the food is coming in, if the kitchen is not ready, the food is not going to stay in the garage. So our body is not built that way. It will come and then it will light up the fire and the food will be prepared, but then the traffic lights are not on, so there will be a traffic jam. So that's why the peripheral clocks, actually, they have a clock, but they also respond to food. And the food tells them when to time their activity. So when we travel, our lighting changes and our food time changes.
Yes. So you kind of are changing gears here, and I think it's very important to point out for people that aren't familiar with this. So we've been talking for some time about this master regulator clock, in the suprachiasmatic nucleus region of the brain, and how light is what sets that clock, what regulates that clock, and in turn those are regulating a wide variety of different physiological processes. But then you just mentioned something very important, and that is that in addition to that clock, there are other clocks, for example, in our liver, in our muscle, in our heart, that also are regulated. But your research, and maybe we can start to talk about this, they seem to be regulated by something different, by when you eat, by when you take in food. And I know for myself, I've known about the circadian clocks and in the liver and how they regulate metabolism. I know that, we're most insulin sensitive, during the early morning hours and most insulin insensitive in the evening. And so I've always tried to not eat too late because I'm like, I don't want to eat this high carbohydrate meal when I'm the most insulin insensitive. I mean, it doesn't make any sense. It's much easier for me to do that in the winter months when it's gets dark earlier. I find it more difficult when spring and summer occur because it's like lighter out. I'm working later, and therefore I eat later. But so let's talk a little bit about how food regulates these clocks and these different tissues.
So a few years ago we started looking at which genes are regulated by clock in different organs. So if we look at liver, there are somewhere between 3,000 to 5,000 genes that turn on a certain time of the day or night. And so that's around what? A lot of genes. So that's almost 30% of expressed genome or whatever. But what is interesting is we said, well, we did a very simple experiment where these are done in mice. So the mice, night-eating mice usually eat during the nighttime. Then we asked, well, there is a master regulator in the brain that's telling the rest of the body when the timing is, when it's day or night. And let's give mice food in the middle of the day, just like the shift workers work in the nighttime, they eat. If we do that, then what happens to the liver? There are all liver genes that cycle, that take the cue from light-dark cycle or from the food. Because we have these two groups of mice, both groups of mice are in the same light-dark cycle. One group eats during day, one group eats during night. If the liver takes cue from light-dark cycle, then all cycling genes should be identical in two groups. If the liver clocks take cue from eating time, then the day-fed animals will have a different clock than the night-fed animals. And that's exactly what we found, that even though the light-dark cycle was the same for both animals, the liver clock responds to when the mice ate. So the day-fed animals had the same 3,000 genes cycling. The night-fed animals had the same 3,000 genes cycling. But now the genes that were turning on during daytime, in the day-fed animals, now they turn on at nighttime in the night-fed animal. So that means the time when we eat tells our liver clock when to turn on the genes and when to turn off. The light has very little impact. We cannot say no impact, very little impact on these cycling genes in liver. So that experiment has been replicated now, and what we are learning is almost every organ in our periphery outside the brain kind of follows when we eat. So then what becomes very important in the daily life is the first sight of bright light and the first bite of food. Mm-hmm. Those two determine how our body clocks work. So now we are working on how this timing of eating or timing of light affects our health in general.
And you have a certain term, I guess, I don't know who coined the phrase, but it's called time-restricted feeding.
Yeah.
And there's been experiments that you've done in mice where you've fed them various types of food, high-fat diet, high-sugar diet, or normal chow diet, and you've restricted their time feeding, during the night, the mouse day hours, which is actually the night because they're nocturnal. Yeah. And you found, some very interesting things. So can you talk a little bit about those findings?
So what we have seen is when we, in experimental animals, if they don't have a clock, then their metabolism goes really weird. So just like I said, a metabolism works like this traffic signal in downtown, and if they're not timed properly, then there'll be disease. Similarly, for a very long time, we knew in the field that mice that don't have circadian clock, because they lack a gene or have a mutation, they have various metabolic defects. They have obesity, diabetes, cardiovascular disease, etc. We also know people who do shift work for a very long period of time, they are also highly likely to get metabolic disease, cancer, accelerated aging. So there was this idea that clocks are important. If we don't have a good functioning clock, then that's bad for us. Then we went back and asked, okay, in normal circumstances, what are the conditions that can actually break down our clock? And what we found was, when mice are given high-fat diet or any unhealthy food, then the food itself breaks down their clock. So they actually don't have a good eating, fasting rhythm. So the mice eat throughout day and night. And we knew that high-fat diet and high-fructose diet, high-sucrose diet, all of these diets that are used experimentally in laboratory conditions give rise to all this disease. And people always thought it's what and how much the mice ate that determined the disease. But what we found is Well, these mice are also not eating at the same time. So maybe when they eat also matters. So we did a very simple experiment where we took two groups of mice, completely identical set of mice. No genes were changed, no drugs were given, and one group of mice ate whenever they wanted to eat. And to begin with, we gave them a high-fat diet. So they're getting somewhere between 45% to 60% of their food from fat, calories from fat. So that means it will be equivalent to humans eating all of their food from cheese, nachos, ice cream, or Western diet.
So they're getting fat and sugar.
So they're getting fat and sugar, all this stuff. And then the other group got the same number of calories and the same type of food, exactly identical food. But they had to eat all their food within 8 to 12 hours in nighttime. So in some experiments, we have done 8 hours, 9 hours, 10 hours, 12 hours, like that. And the most surprising thing means this is something that lot of laboratories around the world do. There are 11,000 papers showing high-fat diet causes obesity. And we said, okay, so now we control for time. So since time was restricted, calorie was not restricted, so that's why we call it time-restricted feeding. And surprisingly, the mice that ate for 8 to 12 hours, they did not become obese, diabetic, and they had a normal liver function, they had normal cholesterol, etc. And then in the next set of experiments, we exposed these mice to high fructose diet, high carb diet, high sucrose diet, all kinds of diets, either ad libitum whenever they can eat, or they have to eat within 8 to 12 hours. And in most cases, we see the time-restricted feeding has huge beneficial impact. Even when mice eat standard diet, normal chow, which is supposed to be very healthy, and mice actually eat most of their food, nearly 70% of food during nighttime. They eat a little bit during daytime. But if they completely restrict that to 8 to 12 hours, then their muscle mass goes up, their fat mass decreases, and they're more coordinated. So if you put them on a rotating drum, then they coordinate on the rotating drum for a long time. So the bottom line is this time. So in these experiments where we kept what and how much they ate constant, the only thing that we changed is when they eat, then we see this huge beneficial impact. And that correlates with very robust clock in the liver and in other metabolic organs. And why this is important is two things. One is many of us have really bad lifestyle. I won't say bad, but we don't have much control over what and how much food we eat. As soon as we get out of our home, all the food we eat outside, we have very little control over it. So the only control we have actually is on our time. So that's why we think this can be a good entry point to a better living by controlling time. And then the second thing is, it also doesn't take away this idea that nutrition doesn't matter, the quality doesn't matter, because even in our high-fat fed mice, we don't see they completely become normal, just like the normal chow-fed mice. They're much healthier. So to have better health, you still have to change what and how much you eat, but timing becomes much easier to manage.
So these, you just covered like so much. This experiment that you did right here, this publication, even before, you've gone on to some small human studies, but this convinced me to do a time restricted feeding schedule because, well, for a couple of reasons. But so just like to reiterate, these mice that were fed a high-fat diet, they fed the same amount of calories, but they that ate during their waking hours, so for mice, which was night, within I think it was 12 hours, they had 70% less fat mass.
Yeah, so they had 28% less body mass total, and that change in body mass is mostly due to fat because they had 70% less fat.
That's amazing.
Yeah, that's really
right there. And they're eating the same crappy food, but they're eating it when their liver can process it the best. When they're able to regulate their blood sugar, when they're able to oxidize fats, things like that. So that was really cool. And then the second thing was I eat a very, very health conscientious. I try to, get a wide variety of vegetables and fruits and good fats and, so all that stuff, omega-3s. But I'm always trying to find more the low-hanging fruit to sort of delay the aging process in a way, or become, as optimal as they possibly can. So the mice that were fed a normal chow diet, high in fiber and all these things, vitamins, you said they actually had more lean muscle mass.
Yeah.
That is very interesting because for me, it's much easier to lose fat than it is to gain muscle. It's difficult to gain muscle, and as you age, it becomes even more so.
Yeah.
And muscle mass is very important. It protects you from frailty, things like that. So any ideas as to how just restricting but we should probably talk about what starts that clock.
Yeah.
What food is it? It doesn't necessarily have to be a calorie, right? It can be black coffee or something like that. But anyways, any ideas as to what is allowing you to keep on more muscle mass?
Yeah, so that actually is a big mystery because in the first series of experiments, we are essentially reporting observations, what happens. The reason why we looked into muscle mass is initially we thought that these mice, when they're going through such a prolonged period of fasting, in some experiments they're going for 12 to 16 hours of fasting every day. And many people think that when you go through this prolonged fasting, you would lose your muscle because the protein gets used to make glucose. That's why you measure lean mass. And surprisingly, we found that the lean mass actually increased, whereas the fat mass decreased. That was a big surprise. That's what we reported. But we haven't looked at exactly why the muscle mass increases. But what we are seeing recently is there is some correlation. Other people have published recently that nicotinamide riboside, this is a precursor for NAD, if that is given to mice, they also gain muscle mass or they maintain their muscle mass. And this nicotinamide riboside is converted to NAD, and increased amount of NAD is always better for any cell because that is the precursor to the energy currency of the cell, that's ATP. And what we are seeing is in many of our mouse experiments, we see the NAD level actually goes up slightly. So this is a natural way to boost up NAD level, not only in muscle, in almost every organ. So I think that might be one of the many different reasons why we're gaining muscle mass. But we can't explain with the current data why they gain muscle mass.
Okay. Can I ask you another question?
Sure.
So I do know that there are some genes, by the way, that are involved in nitrogen balance that are regulated by circadian rhythm. But that's so. I also remember in your paper, I don't know if it was the same paper or a different one. I think it was the same paper, but you also found something very interesting, and it's kind of along the same lines here, and I'll tell you where I'm getting at. But you also found that animals that were fed during a 9-hour period had improved endurance, not improved muscle strength, but improved endurance. And to me, when I read this, I thought, oh, well, if you think about endurance is aerobic, it requires aerobic respiration, which means it requires oxygen, which means it requires mitochondria, because mitochondria are what make energy in the presence of oxygen. So have you thought about looking, and this kind of goes along with your NAD hypothesis, but have you looked at mitochondrial biogenesis, mitochondrial function?
So the endurance is a very interesting aspect because we see that only when mice eat for 8 to 9 hours. We don't see that improved endurance when they eat for 12 hours, although the body weight is maintained as 9 hours. So this was interesting. So that's why, as you pointed out, clearly mitochondria might be playing a role. And in fact, in liver, we do see increased mitochondria volume and increased endoplasmic reticulum volume. So ER is also, ER and mitochondria kind of work together. That's what we are learning these days. So the mitochondria volume increases. Another thing is we do see less damaged mitochondria in liver when they eat only for 8 to 9 hours. Second thing is this mitochondria effect is not restricted only to liver. We do see increased mitochondria volume in brown adipose tissue. So in brown fat, as you know, these mitochondria help kind of dissipate heat, really literally burning the fat. So at least in 2 different organs, we have seen increased mitochondrial volume. That correlates with increased level of PGC1 alpha that's involved in mitochondria biogenesis. So there is all these links that we are seeing, and that are also giving us clue where to look for the mechanism. For example, why PGC1 level goes up and what triggers that to go up.
Well, this actually kind of leads me into another area that I wanted to cover. So before we go into the flies and humans, and that is, I think people may be confused by this time-restricted feeding, which is essentially, feeding within our active hours, the daylight hours, and intermittent fasting. So there's obviously some overlap between the two, because if let's say you're feeding within a 12-hour period, so you wake up, you have your first sip of coffee, that starts your clock. All right, that's it. So 8 o'clock, then you better stop eating by 8 p.m., right? That's for 12 hours.
Yeah.
Then from 8 p.m. all the way till 8 a.m. the next morning, you're fasting, right? You're not getting any energy.
Yeah.
So in some ways, you're getting a lot of, there are some overlaps between this time-restricted feeding and intermittent fasting, for example, which has been shown to increase ketone bodies like beta-hydroxybutyrate, which I know you've also shown restricted feeding does increase that as well. It takes around, I think, 10 to 12 hours for your liver glycogen to deplete, and fatty acids get immobilized. They go to the liver, you start to make beta-hydroxybutyrate and other ketone bodies, which then get transported to other tissues and are used for energy in the brain, or they act as signaling molecules, which Eric Verdeen at UCSF published. And there's lots of, can you first of all differentiate for people like the difference between intermittent fasting and time-restricted feeding? Like what are the main differences, and maybe what some of the similarities are?
Well, both of these depend on this idea. The commonality is this prolonged period of fasting. When I say prolonged, that's usually longer than 6 to 8 hours, because that's how long it takes for glycogen to deplete, or maybe the fatty oxidation to begin, so that we begin to use some of the fat. And you also pointed out ketone bodies and beta-hydroxybutyrate. Those are also produced maybe after 8 to 10 hours. So the bottom line is this: that is, when we eat, we have a type of physiology where we're using glucose and we're driving some bodily function. And at the same time, we may be also damaging some cellular components because of all the reactive oxygen species that we generate during eating, during metabolizing all of this. So all of these have to be repaired, and for some reason, we do not know why, the repair mechanism happens only during the period of fasting. And during this period of fasting, we switch to a different kind of metabolism. Just like you said, our primary energy source is not the readily available glucose from food anymore. It has to come from different sources. In some cases, it can come from a little bit of protein, that's gluconeogenesis, or from fat oxidation, or just like you said, ketone bodies. So these things, this physiology, the fasting physiology, we are just seeing the tip of the iceberg of fasting physiology. We're just learning about a very few molecules. We don't know what happens to a lot of signaling molecules, how the mitochondria actually repair during fasting. Is it actually necessary? To why some repairs happen only during fasting? Why can't they happen when we're eating? So all of these questions are out there. But what is common between this intermittent fasting and time-restricted feeding is this fasting physiology that we are beginning to understand. The reason why we coined and used the word time-restricted feeding is we are not restricting calories, at least in experimental animals. So in that way, the intermittent fasting came from caloric restriction and every other day feeding. that had a serious component of caloric restriction that many people thought is difficult to achieve. So that's why we stayed away from the word caloric restriction or fasting, and we used the word feeding because people may have a positive attitude towards it. Other than that, I think the idea of fasting is ingrained in evolution. Just like in circadian rhythm, the animals have access to food only during their awake time, which can be less than 12 hours. And also for diurnal animals, which are hunter-gatherers, the only time actually they have to hunt is twilight time. Because if you ever go to savannah or any of the African countries where there is still wild animals, or if you go to even a zoo, then you know that animals are not active in the middle of the day. They're mostly active during morning and evening. So in nature, animals actually have only 2 chances to eat, and the rest of the time they're fasting. So this fasting physiology is a very natural response to repair and rejuvenate. And in time-restricted feeding, we're kind of exploiting, or we're kind of bringing back that primordial physiology that's ingrained in our genome, that our genome has to respond to that fasting on a daily basis. At the same time, it syncs with another aspect of the genome, that is, it helps us stay awake for 10 to 12 hours and to reduce our energy level and go into a sleep or less active state for the rest of the day. So in that way, it brings back the primordial rhythms in our physiology, metabolism, repair, and rejuvenation, whereas intermittent fasting actually helped us to learn various bases for this fasting physiology.
That makes sense. I guess also what I was wondering is, if you think about it, like, so the minute you start your metabolism clocks in your liver, for example, the minute you start those metabolism clocks by your first sip of coffee and breakfast, the clock's ticking and you're insulin sensitive. You're going to be able to take glucose up into various cells after you eat, and then once you get past that time, so you're now 12 hours out, you're not going to be as insulin sensitive. So let's say someone that is doing intermittent fasting and they wake up at 6 o'clock or 7 a.m., they have coffee and breakfast, a big breakfast, and they're done. So then they fast for 12 hours. So now it's 7 p.m., maybe 13 hours, 7:38 p.m. They've been fasting all day, so they're getting a lot of the activation of some of these stress response pathways like AMP kinase, and they're making some ketone bodies, Krebs, all these similar pathways are being activated that time restricting feeding also activates. But then they take a big meal at 7:30 or 8 p.m., so 12 or 13 hours after they've already set their clock. So now, in theory, then I don't know if this is true or not. Maybe the intermittent fasting changes some of this, but their liver wouldn't be, it wouldn't be working as well at that point. Or do you think that just because they were fasting all day, that may change some of that and allow them to then eat this meal and it wouldn't have such a negative effect?
Yeah, so that's a very interesting question that we get many times, and we're thinking of addressing that. It's very hard to do that in experimental animal models, because if you give them 2 meals, they reduce their caloric intake. But it's possible to do. But here is something that came out only in the last 3 to 4 weeks. You mentioned early in our conversation that insulin sensitivity is not the same at the end of the day. And the question is, if you fast enough during the day, is your insulin sensitivity is good enough, as in the morning? Then everything equalizes, at least insulin, which is a big thing in metabolism, if it is same. So recently what we're finding is, actually, this smoking gun came almost 10 years ago, when people who were doing GWAS studies to find whether there are mutations in given genes that make us more diabetic or obese. Surprisingly, people thought that, okay, so you'll find some genes that regulate metabolism, right? So that's the common sense. But then the big surprise was they found melatonin receptor as one of the top hits.
Wow.
and some of the clock genes like cryptochrome as in the top 5 or 10 genes. It was not only in one study, in multiple studies they found it. So there was the smoking gun. What is melatonin doing with this obesity diabetes? And recently, what is interesting is people are finding that melatonin receptor is present in pancreatic islet cells, beta cells. And melatonin receptor, when it's engaged with melatonin, it signals and it inhibits insulin secretion.
What?
Yeah,
Really?
yeah. So it just came out like 4 or 5
So
weeks
you know that I've always wondered, because like most of the melatonin in the body is actually made in the gut, right? So your tryptophan gets converted in. So tryptophan from dietary protein gets converted into serotonin. That's converted into melatonin. This is happening in the gut, and I've
No,
always
serotonin
wondered.
goes to pineal and then gets into...
It happens in the gut and it also happens in the brain. There's two separate genes that do this.
Yeah.
And what's really interesting is I don't know what melatonin. Why are we making it in our gut? So I'm wondering if it's somehow signaling to the pancreas.
Yeah. So this is completely new, and so that's why now it brings up. Now it helps us to connect this dot that people have. For last 35 years, clinicians know that the insulin sensitivity is very different between day and night. And then the GWAS, the human genetics people came and said, yes, there is some smoking gun with melatonin. And now finally we are saying, yes, melatonin receptor can actually inhibit insulin secretion. So in that way, having an evening meal, maybe with candlelight dinner, is not a good idea because you have less light, so you have more melatonin, and that can inhibit.
I have to get that study.
Yeah.
Very, very interesting.
So that's one case where we might think that late night, even if you control for calorie, the same calorie taken in late in the night versus early in the evening might have different effect. In fact, there was one study that came out from Spain 2 or 3 years ago now, showing that in a weight loss trial, they actually found, although everybody got the same diet, they were controlled for activity. Clearly, there are two groups of people. One group lost weight significantly, a lot of weight loss, and the other group lost moderate amount of weight. And when they did post hoc analysis to see what is the difference, the only difference they found was the group that lost weight, they actually ate their lunch. It is Spain. In Spain, people eat lunch at 3 o'clock, right? So they ate lunch earlier, whereas the group that did not lose weight too much, they ate their lunch later. So that is another piece in the puzzle saying that late night eating might actually prevent weight loss.
Right. And you've now, you've translated some of these findings into some human trials using this smartphone app that you've developed.
Yeah.
So that's kind of neat as well.
Yeah. So one thing was we wanted to see when people actually eat. And in typical nutrition studies, people are asked, when do you eat lunch, breakfast, and dinner? But that doesn't capture really, they're all snacking and everything. So that's why we thought how to capture when people eat in a very evidence-based manner. And we thought if we ask people to take a picture of their food, then the picture will speak a volume. It will have every single component. They may not have time to describe everything on their plate, but will capture that. It will also have the timestamp. So the whole idea was to see, when do actually people eat? Are there a lot of people who eat like mice do? They nibble throughout the day and night. And if they actually eat until, say, for more than 12 hours or 13 hours, then they are the ones who may benefit from time-restricted feeding. So when we did this experiment, when we started this project, we thought that everybody we asked, they will say, yeah, I wake up, I have my first sip of coffee, and usually I eat all of my food within 12 hours. So we're very discouraged to hear that. But then we carefully selected people who don't do a shift work, so they will not have to work in the night time. That's why they're changing their eating time. And they're also not on any medication that will change their hunger or satiety. So we took really healthy people from San Diego area because we live here. And they just had to take a picture of their food. And that way it was also less stressful for them to enter what they ate and portion size, etc.
Way better compliance,
Yeah,
I'm sure.
there's only 3 clicks, because if you think about it, open the app, take a picture, and then press the save button. And the optional was they could actually describe what they ate. But we found very few people actually described what they eat. So that means just typing that on your left hand when you're eating is not a very pleasant experience. What we found is out of these 156 people, nearly 50% people eat during 15 hours. So that means between their first bite, non-water bite, to the last non-water bite or sip in a given day is around 15 hours, which is, some people think, that's normal, because if they start their first sip of coffee at 6 o'clock in the morning, and then after dinner they're watching their favorite show, and then had another glass of wine or chips, that can go up to 9 p.m. But then we asked, well, in mice, we can actually take away food and impose time-restricted feeding. We can't do that for humans. They have to be self-motivated. So we asked whether it's feasible for some people to at least restrict the time. So we asked 8 of them to see, they were eating for 14 hours or longer, and they were a little bit overweight. So we asked if they can eat within 10 to 11 hours. And we said, we are not going to ask you to change what and how much you eat. The only thing you have to do is select your own time, depending on what time you go to work or what time you come back. Select your own time interval of 10 to 11 hours and try to stick to it every day, even on the weekend. And surprisingly, all these 8 people, they self-selected their 10 to 11 hours, and they stuck to it for 16 weeks. And at the end of 16 weeks, they came back. We saw that they had lost around 4%, 3.8% body weight within the 16 weeks. They didn't have to do too many. They didn't have to read labels. They didn't have to type portion size. But then when we asked them, why did you do it? What is surprising is they said they slept better and they felt more energetic in the morning, and that's why they did it. And since they didn't have to count calories, it was also good. But what is surprising is, in mice, if you do the same experiment, mice will chow down. They will eat the same number of calories as when they have free access to food. But in humans, these people in our study, they actually ate 20% less calories. Even though we asked them to reduce their time, they ultimately reduce their calorie. But if you think about it, this is a much better way to manage their diet than to count calories. So in some way, the study is inconclusive to say whether time restriction alone was beneficial for weight loss. Mm-hmm. But what it showed is the feasibility that some people can time restrict, and that can be an indirect way to reduce your calorie. And since we're collecting picture of every single food, we can also ask another very simple question. What is the time of the day when people are more likely to eat certain type of food? As you can imagine, we found people drink most of their coffee, 70% of their coffee, within 4 to 5 hours interval in the morning. And people eat 70% of their alcohol in the evening 4 to 5 hours. So now imagine if somebody is time restricting to the daytime, then he or she is more likely to lose on alcohol. So in that way, that also improves the quality of diet. So since we humans eat different type of food at different time of the day, depending on which interval we choose, may indirectly result in change in nutrition quality and to some extent quantity.
And what about cutting out like the ice cream and desserts?
Yeah, so we most of the reduction in calorie was due to reduction in late night snacks and after dinner ice cream, dessert, and alcohol.
So it's a really cool idea, Satchin. I went to your website last night, mystcircadianclock.org. I really like the website in general. There's a blog section, the section explaining, a lot of the science behind circadian clocks and everything that we've talked about today. There's some presentations of you there. I mean, it's just really great website. But I also signed up for the app because I am now on a time-restricted, feeding schedule.
Yeah.
right now I'm doing 12 hours. I would like to try the 9 hours to see if I can get any endurance benefits, which may possibly be mediated by beta hydroxybutyrate, because that's been shown to affect endurance. But anyways, so I went up and I signed up. It's really simple. You go to the website, you click. Maybe you want to explain. I clicked sign up or something, and then
it asks you a very simple question. So the whole idea was, if you think about it, this branch of science, circadian rhythm or circadian research, came out of curiosity. So we actually don't have a traditional medical school department which will take our results and translate to public, or it's not even in the public health curriculum, because circadian rhythm is such a new field that's not there. So we thought
so important. And so it's like disrupted by modern day society. I mean, it's like I'm going to be talking about this now, and so I'm very excited. It's definitely important.
No, what we are saying is that if you brought up this modern day society. In modern day society, light is an enabler. So light enables us to stay awake throughout the night. And then we have 15% of workforce who does night shift work, and they are the ones who actually enable the rest of us to stay awake. They are the ones who are actually driving the truck, who are in the emergency department, they are food prepping, they're doing all these other service jobs, and they enable the rest of us to stay awake. And then in a way, within the last 100 years, we have gone tremendously from a very natural day-night cycle to 24 hours light cycle. And that's the biggest disruption we're seeing. And that biggest disruption leads to all types of chronic diseases that we see in modern days. For example, now, out of the top 10 causes of death, if you look at the top 10 causes of death in industrialized country, the top 5 or 6 are chronic diseases. And we know circadian disruption can lead to those chronic diseases. So now the question is, if we can do very simple adjustment to our lifestyle, can we prevent this chronic disease by X number of years? So to get to that, we need two different information. One is, what is the extent of circadian disruption? When do people go to sleep? When do they get up? When are they eating their food? And when are they exercising? So if we can capture what, when, and how much people eat, sleep, and move around, then we have a very complete, nearly complete picture of somebody's lifestyle that will be highly useful for circadian rhythm research, for your primary care physicians, and also for public health and epidemiology. So that's the first part of the goal, to collect what, when, and how much people eat, sleep, and move around on a daily basis for at least a week or 2 weeks. So we'll know how is their lifestyle during weekday and weekend. And then in the second phase, we can give some health information, because many of the health apps are more geared towards losing weight, doing one thing, and it's very difficult to really guide people to do these 3 different things: eat, sleep, and move around on a timely basis. And it's a challenge. We're experimenting. So we are actually hoping that some people who are signing up, they will give us feedback how to improve our science and also our education and our app, so that we'll see the benefit. In the second phase, they can self-select, just like you said, you're going to self-select 12 hours, maybe 9 hours. And when you self-select an interval and enter all of your food, and sometimes even if you forget once in a while, that's okay, because we account for that. We have some algorithm. And then we want to correlate whether this 9 hours is beneficial to you or not, or whether some people actually get the same benefit with 12 hours. Some people might go to 8 hours or 9 hours. So those information will come in the second phase that goes from second week till 16 weeks. And then if you want to continue, we have many users who want to continue for a year or 2, and they want to see how seasonally their eating pattern, sleeping pattern changes, that they can have the data, and also we gain from that data research gains by taking that data and seeing what is the pattern in general public.
Also, it's nice to feel like you're contributing to research. So in addition to this My Circadian Clock app, which is it's on the App Store, it's Android,
and also in Android.
And so in addition to that app, you can you can sync with other fitness data like MyFitnessPal, or so because you're going to be measuring all these other health parameters.
Yeah.
I heard you mention something also about a light sensor thing or some.
Yeah, so I wear a light sensor that senses light. But let's go back to the Health Kit and Google Fit. So most phones now have, if it is an iPhone, it has something called Health Kit that comes with your operating system. So it's already in your phone. And that phone, that Health Kit app is sensing every time your phone moves. So it's almost like it's measuring your movement just like the Fitbit does, and it stores that data. It also stores many other kind of data if you want to store. For example, your own information about your body, your height, weight, age, etc. And it can record up to, I think, 70 plus different parameters, depending on if you're using a special app or even the same app. If you say what you weighed, what are the composition, it can store that. But what is interesting is that information is not on the cloud. So it always stays on your phone. If you lose your phone, you lose that data. But what you can do is, if you want to share what is in your Health Kit or Google Fit is very similar to Health Kit, but it runs on Android. What you can do is you can sync that data with an app like My Circadian Clock. So any information that you store in Health Kit gets shared with My Circadian Clock. And My Circadian Clock can also send some data back to Health Kit. So it kind of acts as a hub. So similarly, various apps like MyFitnessPal and then Noom and many diet apps, many exercise apps, they also deposit their data to My Health Kit or Google Fit. So it's kind of a data exchange hub. So if anybody has any app where they are measuring what, when, and how much they eat, sleep, or move, or any blood parameter, any other health parameter they are interested in sharing with researchers, they can sync that to Health Kit, and then Health Kit gets synced with My Circadian Clock, and we can capture all that data and we'll analyze.
Very cool idea. I'm looking forward to contributing to that. But I kind of wanted to
talk about the light.
I want to talk a little bit about your. So you've got so many awesome. So much awesome research coming out of your lab. There's also the heart rate, heart rhythm studies that you mentioned. So you mentioned earlier the chronic diseases. If you look at in the United States or industrialized societies in general, the number one killer, people die the most of heart disease, some sort of heart disease. So, and obviously lots of different things regulate our susceptibility to heart disease, and metabolism, obesity. But you found some very interesting findings doing time-restricted feeding in fruit flies. So can you talk a little bit about that? I'm very interested.
So fruit flies are used in science for many years, and they have a short lifespan. They stay alive maybe 9 to 10 weeks max. So it helps us to figure out whether any intervention like time-restricted feeding will have any positive or negative impact on health span or healthy lifespan or longevity, etc. So what we did was, again, a very simple experiment. We took fruit flies and we gave them food only for 12 hours during daytime, because fruit flies are diurnal animals. They fly around during daytime, eat, and then nighttime they sleep. Or they had access to food for 24 hours. And we measured that they were eating the same amount of calories, and they were also moving around the same distance. So inside these bottles, they could fly back and forth. And at 3 weeks of age, their heart is very healthy. It beats rhythmically. And although the fly heart is not like human heart, they also have...
Those people are probably shocked flies have a heart.
So their heart is very similar genetic program. In fact, many of the genes that are now known to be necessary for heart development in human were discovered in flies. And vice versa, there are many diseases in humans. Those are now put into flies to see what do they do in the heart. So it's a very interesting model. Just like humans, the fly heart also becomes weaker with age. So by 5 weeks, the hearts don't beat rhythmically. They have a little bit of arrhythmia, and then they have the same dialysis, so the heart gets dilated with age. The beat-to-beat distance also becomes very irregular. So what we found was when these flies eat only for 12 hours, they don't develop that arrhythmia as quickly as the normal flies do. So they're protected from this heart disease. Then we said, okay, so if we introduce time-restricted feeding later in their lifespan, then what happens? Because one thing we could not do in mouse study, because mice live for 3 to 4 years, we could not introduce time-restricted feeding later in life. But in flies, when we introduced later in life, they were also protected. The arrhythmia reduced in flies. And when we gave high-fat diet to flies, they also produce arrhythmia and many heart conditions that we see in humans, and those were also protected in flies. And what is interesting in flies, we also saw the flies sleep better when they eat only for 12 hours. So by 5 weeks, flies actually are just like very old people. They have fragmented sleep at night time, and they're sleepy during daytime, and that is completely prevented by time-restricted feeding. They have a good night's sleep, they're very active during daytime, the heart pumps nicely.
Wow. Did you measure heart rate variability? Or?
Yeah, so we did heart rate variability. So there are 7 different parameters we measure.
And that improved?
Yeah, so all those 7 parameters improved to some extent. And when we introduced later in life, they also improved. That was the surprise.
Do you know what or why?
Yeah, so what we found is, again, one connection was back to mitochondria. What we found was that mitochondria were healthier in the sense maybe they were not producing as much reactive oxygen species.
The mitochondria in the cardiomyocyte, in the heart cells?
In the heart cells.
Okay.
So we took the heart cells and did gene expression profile. We looked at all the genes. What we found is there was a big cluster of genes whose expression actually reduced, and those are from the electron transport chain. So that implied that maybe they have less reactive oxygen species, or maybe reduced activity of ETC, electron transport chain, is beneficial. So then to prove that, we actually knocked down few components of ETC, and those flies also have better heart. So that was one thing that we found. Second thing that we found is proteostasis, or protein folding, is necessary, and in many other organisms people have shown that in different components of protein folding machinery. But here what we found is there is a new protein folding, very newly identified folding machinery called ATP dependent. It's a chaperonin complex. 8 different components form this barrel-like structure to fold proteins. And this requires energy. And that CCT component has been shown to be important for various cytoskeletal protein folding. And it makes sense for heart. And in fact, in humans, there is a point mutation in one of these CCT component that has been shown to predispose to some heart disease. So it's a beautiful story where we found both mitochondria and this protein folding machinery are necessary for this time-restricted feeding beneficial effect.
And this time-restricted feeding was 12 hours?
That was 12 hours because flies are different from us. They don't have thermoregulation. So when they fast for a longer time, they cannot. They can't tolerate that.
Oh yeah, they'll get cold. Okay, yeah. So some of these phones also measure heart rate variability.
So it'll be
interesting. Very interesting, because that's like, that's something that's supposed to be very important for having a healthy heart. But if you don't mind, again, it's another reason why this time-restricted feeding, it just seems like everyone should be doing it. It's an easy lifestyle adjustment. I mean, easy enough. You have to be disciplined to some degree, but it's easy enough that you can do. It's like, by the time it's 12 hours later, all right, that's it. You got to make sure you get all your food, your whatever it is that you consume within that 12-hour span. But something I did want to shift gears, if you don't mind.
Sure.
Another organ, the gut, because it's another very area of interest of mine. And you recently published that the gut microbiome, so we'll quickly go through this, the gut microbiome is also, bacteria are living organisms, and they are also on a circadian rhythm.
Yeah.
And what I found very interesting from some of your recent work is that you showed that different bacterial species within the gut, at least in a mouse, also seem to have more of these species during certain times of the day and less during certain times of the day. And this was different in obese animals versus non-obese animals. Or that is very fascinating. Can you explain that?
Yeah, so bacteria, they just like us, they need their own niche. They need their own pH, temperature, nutrition, and all these factors. And now you can imagine that when we eat, we kind of change the gut environment, the content of the gut slightly, so that some bacteria may find it easier to grow, and then some other bacteria may find it very difficult to grow in that condition. So what happens is when we eat in a time-restricted fashion, we have a very fixed eating and fasting interval. Then during eating, that environment will promote a certain set of bacteria to bloom, and then the other bacteria kind of become quiescent, quiet. And then after a few hours of fasting, then the second set of bacteria will bloom. So in that way, what it helps is, it helps to nurture a wide variety of species to cohabit in our gut. And they also function at different time of the day, during the daytime. So for example, when we are eating, we need a lot of bacteria to break down starch and fibers, complex carbohydrates, and also conjugate some bile acids and etc. So we need different players to do different things throughout day and night. And by time-restricted feeding, this alternating two different types of environment, one where there is a lot of food and the pH is very different, versus one when there is fasting, there is scarcity of food, and the pH is very different. By alternating between these two different environments, we promote this diversity inside the gut. And why this is important is we actually don't understand why, but research from a very different field, from gut microbiome field, they are now finding is it's much better to have a more diverse microbiome in your gut than to have only one or two very simple species. So in some way, by having the time-restricted feeding, it promotes the diversity. There are a few interesting things that we found, which we are still working on to figure out why it happens. For example, mice that had time-restricted feeding, although the bacteria could break down the complex carbohydrate to simple carbohydrate, for some reason that simple carbohydrate could not be taken up by the gut. It actually went out in the poop. And that was a surprise, because usually simple carbohydrates are the ones that get absorbed. So we think that this compositional change somehow protects the complex carbohydrate in the upper intestine. So it comes back to the lower intestine where it gets degraded, but we know that the lower intestine doesn't absorb sugar. It's only in the upper intestine. So in that way, by this compositional change, we can completely shift how nutrition is even absorbed into the gut. The other thing that we also found is bile acids, which are emerging as very important players in health. Those bile acids are also better managed with time-restricted feeding, both in the liver and also in the gut. Bile acids are made in liver, and then they cycle back and forth between gut and liver. And bile acids are made from cholesterol. So there is an enzyme in liver that gets upregulated in time-restricted feeding, and that enzyme breaks down cholesterol to bile. So you get dual benefit. You reduce cholesterol, increase bile acid. And that bile acid comes to the gut, and it helps absorb some fat back into the gut. And then it gets conjugated by this bacteria. And for some reason, we don't understand, in time-restricted feeding, they get modified in a different way than in normal feeding. So these are some of the new research directions we are taking. We're trying to understand how this gut microbiome interacts with timing of food, and then changes the compositional aspect of the gut.
It's very interesting. Have you looked at also the short chain fatty acids that they produce, and whether those get taken up by the gut?
Yeah, so
epithelial cells?
we are now, so short chain fatty acids are a little bit difficult to look at because they're volatile, they
Right.
degrade much faster, so those are the next set of experiments we are doing.
Okay, So does this also suggest we should time our probiotic intake? Is there
We haven't looked into that to see whether the probiotics.
is there any data out there that has measured? So, for example, I've measured my own microbiome using a company called Ubiome, which
Yeah.
allows you to send a little fecal sample,
Yeah.
and I've done that a few times. Obviously, now it seems like the time of day is very important. I usually do it in the morning because But it seems like the time of day is very important. But
Yeah,
also,
you can capture the compositional change by time of day.
Is there any data out there that has looked at how the microbiome species change from morning to evening? Like,
Yes, actually in
is?
humans there are at least now 1 or 2 papers showing how the compositional change also changes throughout day and
Take a look
night.
at those.
And when you have jet lag, that messes up the
Right.
compositional change.
Right. Yeah. So,
Yeah.
I think there was some study showing the microbiome gets thrown off, and that leads to obesity or something.
Yeah.
There's just so much, and then of course the shift workers as well.
Yeah.
I mean, they're completely. Their whole system is off whack. It'll be interesting to see whether or not time-restricted feeding can help negate some of the negative effects of
shift work.
work, right?
Yeah,
That'll be
so
very
that's what we're
interesting.
doing now to see whether we can put mice in shift work and then give them food within shift or out of shift. So
Do you have an option on your app that for shift workers so that you could also get some human data?
yeah, so people can say what they, so in the first sign-up routine, and there is a question whether you are a shift worker or a regular worker. And if they say they're shift worker, then we look at their data more carefully to see how they're changing their diet during day shift and night shift, and also all shift works are not the same. Some employers actually put people change shifts every week, every 2 weeks, every 3 weeks, and in some cases every 4 months.
Yeah.
So we'll see all that data. But we are going to introduce another aspect where they can say when they're going to work and when they're coming back, because there are now different variations of shift work. Many of the gig works of flexible shift work is not even fixed. So flex hours is a new trend where people can be called off anytime, and we want to track them more carefully. So that's why we'll have another feature soon.
Okay. And what about, so I know when I filled out the questionnaire last night, I put the city that I lived in. Is it, so let's say I'm traveling and I take a picture of my food, there's a timestamp, are you going to get the time zone? Like,
yeah, so in the new, it always tracks the new time zone, because what happens is, we thought about it. It's a little bit complicated to show 2 different time zones in your home time zone and then in the new time zone. But we'll get to that to see whether we can introduce that feature where you can see your data as if you are in the same time zone, how your body is adjusting. You can see that data. We are working on it. What we figured out is time is a very difficult parameter in apps, because as we are moving, as there is daylight saving time and all these other time changes, completely displaying data becomes a challenge. So that's another half of the challenge.
yeah, no, what you're doing, but you're off to a really good start.
Yeah.
I'm really excited about everything that you're doing. I have so many more things that I wish I could talk to you about, but unfortunately we're running out of time. I know you have another meeting. So you are on Twitter. I follow you on Twitter. What's your Twitter handle?
Satchin.panda.
Satchin.panda is your Twitter handle.
Yeah.
It's
SATCHIN.panda, as in panda bear.
Right. Okay. And your website, mycircadianclock.org.
Yeah.
And that's where people can find all things circadian clock, all fascinating research, you giving presentations, and also they can sign up.
They can sign up. And also, almost every week now, we'll have a blog, either from actually, we are getting a first blog from a user, just like you. This person started before even our human work was done, and he just started voluntarily and religiously did 9 hours or 8 hours time restricted feeding for 7 months and documented everything. So, so that's going up this Friday. So we are very excited.
Yeah, I really like your blog. I was
Yeah.
looking over it last night. It's very informative. So it's a nice job. Well, thank you so much, Satchin. I've been a huge fan of your work for several years, and pretty much all things that I've learned about circadian rhythm, I'd say 80% has come out of your lab. So I'm very excited to have had the opportunity to speak with you.
That's very nice of you. Thank you so much.
I look forward to more research coming out of your lab. You seem to be doing some really interesting things. So
Thank you. Thank you.